If you accidentally touch a hot stove, your hand pulls away before you even consciously register the pain. That reflex happens in milliseconds. But human bodies aren't wired with highly conductive copper. Our internal wiring is made of salt water, lipids, and squishy organic tissue. Water naturally dissipates electrical charge. That makes it an incredibly difficult environment to generate and transmit a crisp, precise electrical signal over long distances. To solve this, the body transforms every single neuron into a microscopic battery. It builds a constant state of electrical tension across the cell membrane, ready to release a spark on demand. The sodium-potassium pump burns cellular ATP to push ions against their natural flow. It forces 3 internal sodium ions out, then pulls 2 external potassium ions inside. Since 3 positive charges leave for every 2 entering, there's a constant net loss of positive ions. This relentless molecular pumping locks the cell at a resting potential of roughly -70 millivolts. The neuron is now a coiled spring, vibrating with stored potential energy. This graph shows how a neuron's voltage behaves over time. The baseline rests at -70 millivolts, but there is a strict biological threshold marked here at -55 millivolts. Neurons operate on an all-or-none principle. Minor chemical inputs from other cells might cause slight voltage bumps, but if they fall short of that -55 mark, they fizzle out completely. But the exact moment the voltage crosses that threshold, voltage-gated sodium channels in the cell membrane are forced to slam open. Driven by the immense tension, sodium violently rushes back into the cell. The internal voltage rockets to a positive 40 millivolts. To prevent a short circuit, an immediate protective reset kicks in, known as repolarization. The sodium channels snap shut, and potassium channels open to flush positive ions back out. This rapid electrical spike and its immediate reset is the action potential, the fundamental single unit of biological data. That initial electrical explosion creates a positive feedback loop. The massive influx of sodium at one spot triggers the next set of sodium channels further down the axon to open. To keep this signal moving fast over long distances, evolution developed saltatory conduction, a process relying on a fatty insulation wrapped around the axon called the myelin sheath. Instead of traveling in a slow, continuous wave, the electrical signal is forced to leap directly across the tiny exposed gaps in the myelin insulation. Right after the spark fires, the cell undergoes a brief refractory period where the voltage drops even below the resting -70. This extreme negative state acts as a physical barrier, making it impossible for the signal to travel backward. The combination of this jumping conduction and the trailing refractory period ensures the electrical spark travels at breakneck speed in a strictly one-way journey. But there's a physical roadblock at the end of the line. Neurons do not actually touch. They are separated by a microscopic 20 to 40 nanometer gap known as the synaptic cleft. An electrical current cannot jump across empty fluid. To cross this void, the spike must be translated into a chemical messenger. When the action potential arrives at the presynaptic terminal, it dumps specialized molecules, neurotransmitters, straight into the gap. These messengers drift across the space and lock into receptor slots on the next cell, instantly starting a brand new action potential cycle. This seamless translation from electrical spike to chemical drift and back to electrical spike links billions of isolated cells into a unified, thinking network. This finely tuned system has a vulnerability, and nature has evolved highly specific countermeasures to exploit it, most notably tetrodotoxin. You can find this incredibly lethal defense mechanism in animals like the pufferfish. The toxin operates with terrifying precision. It acts as a biological cork, physically lodging itself into the voltage-gated sodium channels. With those doors jammed shut, action potentials become utterly impossible, completely shutting down the nervous system. Without that delicate, relentless cycle of chemical tension and electrical explosion, the system goes dark. It is the invisible biophysical engine driving every thought, heartbeat, and action we will ever take.